Martin Kräter is a postdoctoral researcher and fellow at Max Planck Institute for the Science of Light in Erlangen. He has been investigating physical properties of blood cells since many, many years, and he has also run some studies together with his group in long COVID patients and acute COVID-19 patients, and he found some abnormalities in blood cells, their stiffness or different deformability. And this could explain some of the symptoms. And this tool, the deformability cytometry, is something that's not established yet in clinical routine. Hence, this is why he's going to talk about bench to bedside. And we hope, of course, we can translate it very soon into clinical routine. So Martin, the stage is yours. Thank you so much. We will have a Q&A running. And after 20 minutes talk, we can address some of the questions of the audience. Thank you so much.
Yeah, Diana, thank you. Thanks for the invitation. As you said, I'm going to talk about my favorite piece of technology, deformability cytometry. And I rephrased it a bit. So what is it and how can it become useful in the context? First of all, I would like to thank all my team. That is basically what you see here is the team of Jochen Guck. He's the director here at the Institute, and the ones marked in yellow now. This is my team, and we are working towards the use of this technology into the clinics as well. And of course, without them, nothing would have been happened until now. I'm also super grateful to the organizers of that fantastic conference. I have heard yesterday a couple of talks. It's really great opportunity for collaboration between patients, researchers, and clinicians. And what the organization has done is really fantastic. And I think it can't be honored enough by everyone. Before we dive into the topic, I just need to state that I'm co-founder and share owner of the company Riverside. We are selling deformability cytometers for blood cell analysis. And with that being said, I would like to start with the question, what is physical phenotyping and why do we have to do that? I think you're all familiar with such a picture here. You have done it in the supermarket very often, right? What you try is basically, you try to identify this continuum here of this fruit or vegetable, I don't really know. And you try to identify it without any optical methods, right? So because the optical methods are not sufficient enough to figure that out. And therefore, one can draw a simple conclusion, and that is that the mechanical probing reveals information about the pathophysiological process of tissues. And what we have done is basically we have brought this down to the single cell level. So we are not interested in tissues anymore, but on the smallest living objects in our body, and these are cells. And as I'm from the hematology field, I basically brought you a hematological example of this, where we have looked at bone marrow-derived common myeloid progenitor cells. These cells are basically producing the cells, the blood cells of the myeloid lineage, so the erythrocytes, megakaryocytes, but also all the immune cells, monocytes to granulocytes. And I have picked me this progenitor and fully differentiated neutrophil, and I looked at the cytoskeletal arrangements. And as both of these cells have completely different functional state, right? So these cells, they have to survive in the bone marrow, they have to proliferate and produce the blood cells, while these cells have to circulate and defend the diseases. We checked the mechanical properties of these cells. And what you see here plotted is basically the deformation. So basically how much a cell deforms under a certain load and the area of the cell. And you can clearly see that there is a difference in the deformation where the area is almost identical. So you see that the neutrophils are much softer. They deform much more under the same load. And this has impact for their functional properties. You can immediately see here when we facilitate microfluidic channels where the cells, as in the circulation, for example, or in the smallest capillaries, where the cells need to migrate through these capillaries, you can clearly see that the precursor cell with its stiff phenotype cannot enter this channel, while the neutrophil can easily do that. And so one can say that by just this simple test of measuring the mechanical properties of the cells, you can get a clear functional readout and can predict what the cells will do in the near future, maybe even. That this holds true in a diseased state was already published here in an in vitro experiment where we treated blood with different stimuli. So, for example, LPS, surface protein of, for example, bacteria. And what we plotted here is, again, the deformation of the neutrophils over a course of 300 minutes. And what you can clearly see is that after a very short timescale stiffening phase of the cells, so the cells become less deformable, they all increase and become more deformable. So the neutrophils seem to get activated. That's what these drugs do. And then they change their physiological state and become very migrative. So they want to migrate out from the circulation and go into the tissue. That this holds true is something we have seen also in COVID-19 patients, severely ill COVID-19 patients. Again, the deformation of the neutrophils plotted for control people, for COVID-19, severely ill people, and then also for people who have recovered from the disease. Back then, when we published this study, there was basically just the community of long COVID people speaking about long COVID or PCS. And we have seen that these recovered people have still changes present, which are also present in the severe state. What we can say is immune activation of neutrophils can be monitored by the deformation. So you might now ask, what holds us from using it? Well, there are a couple of techniques, from micropipette aspiration, atomic force microscopy over optical stretcher. And all these techniques have one thing in common. They are super, super slow. So you reach 10 cells per hour throughput to 100 cells per hour if you're a very good experimentator. And it also requires a lot of knowledge and experience to operate these technologies. And we tried to bring this to the next level. What we are doing is we're facilitating most modern microfluidic techniques together with ultra-fast imaging. What we do is we send cells through microfluidic channel constrictions where, as you can see here in the video down, where they deform into this bullet-like shape. In the initial state, they are round, and then they get deformed into this bullet-like shape. And as we know the boundary conditions in these channels from the flow, we can basically calculate the mechanical properties of these cells. And what you can also see is that this all happens in a millisecond area. And this millisecond timescale is super important because that leads us to a measurement rate of over 3,000 cells per second. So from 100 cells per hour to 3,000 cells per second. And that allows real blood analysis in the clinical state. What we are doing is we take little as 10 microliters of blood. We send it through our device. We image the cells. We end up with 2 million images per patient, so 2 million cell images per patient in a single measurement. Then we use AI-based algorithms to find the cells in the images. And from that, after finding these cells, we segment the cells. We extract up to 27 different features. These are from the deformation, as I told you, the area, the volume, but also surface roughness and so light transmittance. With all this information and having a picture saved of all these cells, we can basically just facilitate artificial intelligence algorithms, so neural nets basically, to do the classical blood count, for example. Just for you, that you get a feeling. So here you see a thrombocyte, you see an erythrocyte, you see an erythrocyte doublet, lymphocyte, monocyte, neutrophil, and eosinophil. And this can be done fully automatically in our system. But what is it useful for, right? So assume we have now basic manual analysis. Can we detect disease with that? And I want to introduce you to our very unbiased measurement of blood. Why is it unbiased? Well, our machine is just taking images at high frame rates, right? And with these images, we can basically not only detect the classical cells, as again here the lymphocytes and the granulomonocytes and the erythrocytes, but we can also detect much more things or much more events which appear in the blood. Like what you can see here is basically leukocyte aggregates, but also like these kind of microclots. Back then we teamed up with Rasia Pretorius and we got these nice electron micrographs from her. And we looked at whether we see the same pathological states of the cells in our images. Obviously, electron microscopy takes much too long and and a lot of preparation for the cells. And yes, we find these pathological states, right? Erythrocytes with thrombocytes attached, fibrinogen connecting cells to each other, or fibrinogen activated thrombocyte clusters. And even more, we think we also find from time to time endothelial cells or endothelial debris in our measurements. At the moment, and I'm not going to show you now the results of what we have seen in the patients for the lymphocytes and leukocytes, but I want to emphasize the clot detection a bit more. I think it's a very, very interesting topic and something which a lot of patients have in common. And we checked, or we wondered whether these aggregates here are, for example, really thrombocyte aggregates. And what we did is basically we stained the cells with classical surface markers, stained for thrombocytes here in green, and stained also for activated thrombocytes here in orange. And we co-stained with endothelial marker. And what you can see clearly is that this clot contains of activated thrombocytes as well as this, as well as that. And we continued doing that, and we found in the end also clots which only contain of fibrinogen. Then we find clots, fibrinogen aggregates, and other cell aggregates, like for example red blood cells, as you have seen it also in the electron micrographs. Plus, we have huge cell aggregates, which we think are endothelial debris. Furthermore, from time to time, we see like really crystal structures where we yet not sure what these crystal structures are. At the moment, we are trying to add this information to our neural nets in order to make a classification of not only the cells, but also different subtypes of clots, which we have currently operating somewhat in the range of 30 different classes, event classes in blood. And I want to stay with the clots just a little bit, and thanks to Carmen Scheibenbogen, who teamed up with us in order to start looking at aggregates in serum, in patient serum. So she sent us the serum, and we basically, with our features, identified the different clots. We separated these clots into small, medium, and large aggregates. This is arbitrary. We just made up a decision of what could be small, medium, and large. And then we looked at 3 different subsets of people. The first subset is post-COVID syndrome people with ME/CFS, post-COVID syndrome people without ME/CFS, and controls. These patients were obviously defined by Carmen, and we have measured them. And what you can clearly see is that of each kind of these clots, the people with post-COVID syndrome and ME/CFS have elevated numbers even in their serum. Furthermore, as I told you, right, so we have also available this deformational flow in which we operate. And what we can do is basically we can look at the aspect ratio of the cells. So how long are these events over how high are these events? Simply said, we check their X lengths, we check their Y lengths, and we just calculate X over Y. And what the aspect ratio gives you is, as lower the aspect ratio, as more round the objects are, so they do not deform. And we see again that for post-COVID patients with ME/CFS, that also these large clots do not deform as much as the ones for non-ME/CFS or control patients. That means that not only these patients have more of these clots, they also have basically less deformable objects. I just want to say it here, this is all unpublished data, and I'm very grateful that Carmen allowed me to show it here in this conference. In the end, what are we going to do with this information now? Well, first of all, we thought, is it actually useful for anything? And what we did is, we checked other diseases. What you can see here are 5 different disease states, from acute lung injury over respiratory tract infection, Epstein-Barr virus infection, COVID-19, and post-COVID syndrome. And we took arbitrarily 6 different features. We looked at lymphocytes, monocytes, and neutrophils, deformation, and size of each of these cell types alone. And what I plotted here is just in white, when there is no difference to the corresponding control set, and in gray if there is a difference to the corresponding control set. And what you can clearly see is that even though these diseases present in the clinics, specifically these ones here, present in the clinics very similar, they have clear fingerprints of the disease state. Okay, with that being said, we can maybe, and this is something you can say here, for the post-COVID syndrome patient, you see that also the post-COVID syndrome patient have a different fingerprint in only these 3 cell types from these other diseases. And so we hope even that we can get a more sub-classification of the post-COVID syndrome patients, as we heard it now from all our previous speakers, that there are subclasses in the patient cohort. So what are the next steps? What is basically the problem to bring this to a diagnostics approach? Well, the first thing is obviously that you would need to have technology and the corresponding consumables. As said, we have founded a company in order to do so. We have available the hardware, we have available the software, and we have available the consumables to measure those patients and to bring these devices into clinics. What is needed to push this further is obviously that the funding scheme has to change towards basic research. What's currently happening, specifically also here in Germany, is that healthcare-related research is promoted, which I can totally understand. Obviously, it is the case that we need patient care and drastic patient care. But also the basic research needs to come up in order to produce new technology, such as deformability cytometry, to make diagnostic approaches, and based on these diagnostic approaches, maybe subsequent treatments. And then there's another thing. Well, in order to make a product, you always need IP, right? So without any IP application, you will never ever have a product which can be in any case refunded. And IP applications, they also tend to take very long. We are on a good track in that regard. I think this is all covered already. But then there comes the regulation part. And the regulation is something which takes a tremendous amount of time and is not fast, as we have seen it with the emergency regulations during the COVID pandemic. So what you have to do is basically you have to obviously do the preclinical testing. You have to do the clinical investigations. Then you have to do the conformity assessment, and all these paperworks take years to get granted. This is a representation of what all needs to be done in order to get a certificate for such a product. But as it is often more represented in the in the organizations, is like this. So it looks like a super nice process which just goes along this line here, but this is actually not the case. It is very, very complicated and takes a lot of time and effort. Plus, these overviews, both of them, do not include the finances. So basically, the money needed to go through all this process. It obviously needs a ton of legal advice, and we have one problem. Specifically, if you use artificial intelligence, the regulations are somewhat implemented, but not fully clear. And then, as it is in our case, we have consecutive AI packages in our device, and this is yet not considered in such a scheme. And therefore, I guess we have started the process, but to the real application, it will take us definitely a while. And with that, I hope I'm not over time. And I definitely have to also acknowledge some of the funding agencies, which you can see on top here. But I would like to thank primarily all our collaborators from the Technical University Munich, Stellenbosch University, Charité, Uniklinik Erlangen, obviously Riverside. And if you are interested in seeing how Riverside performs and what we are doing, you can scan this barcode and basically register to our newsletter.
Yeah, thank you so much, Martin, for sharing unpublished data and for showing that what this technique is capable of, like differentiating between the different diseases, between COVID and long COVID, and that there's still a lack of funding and legal aspects, and the process is very highly complex. I would like to address now some of the questions we had from the audience. So the most upvoted questions is from Tom. He's saying, how much coordination and collaboration is occurring right now? Having watched the past 1.5 days, it seems there's loads of research getting done in identifying a hypothesis and treatment approaches, but no overarching leadership. So how can we optimize the next steps?
Yeah, that is really good question, and I think there's a lot of things are going on, but primarily leadership and project coordination is something which is in the system not really funded, right? So that is something which is not included in the general idea of the system, and that needs to change. And therefore, it's obviously great that people like you guys are trying to bring the people together in a certain sense, but also you are unfunded. You are funded by the people who are sick, and I think this is not the right way to bring disease diagnostics and treatment forward. What can be done? So it is obviously that we have to put in much more effort and money into coordination of the projects. You know, everyone is trying to collaborate with a couple of people, but there is no way of combining all this knowledge so other than reading basically papers of the other researchers, right? And paper publishing and stuff, that also takes time. And even we have, a flood of papers coming out at the moment on the topic. All these findings were made back in time, right? So so we are not staying on top of the time here.
Yeah, thank you very much. Yeah, that's very insightful. I think there are a couple of fundings nationally that try to foster these collaborations, but there's a huge lack. I appreciate you mentioning that here so loud. So we have another question. What does it take to accelerate the rollout of these diagnostics you have been mentioning? And would there be 100% reliable biomarker?
Well, so I mean, let's start with the second question. There is never ever 100% reliable biomarker. That is something which doesn't exist. I mean, despite the fact that you broke your leg, right? So I think the biomarker that you broke your leg is kind of 100%. But I think we are not operating with one biomarker. We are operating with at least 600 features per measurement, and that makes it, very reliable and very stable measurement. And that is what my hope is, that we find here, not only a diagnostics, but also a subcategorization of patients and therefore informed treatment opportunity. What it takes to accelerate the rollout, well, that's obviously reducing the amount of time it takes to get certified and the needed money to go through all this process, right? From clinical studies to legal advice.
Okay, so it's a question about money and making the processes a bit more less bureaucratic, I guess, right?
Mm-hmm.
Okay. So then there's another question. I don't know if you can answer that. Since these tests are not, the tests you're offering are not in clinical routine yet, are they available to physicians and clinical practice that would like to offer that service?
Well, of course, in principle, in Germany, I would say that this is possible, right? So you would need to get such a device and do the measurements. What we are currently doing is we started collaborating with a lot of people and started to engage in the respective clinical studies on treatment and on these healthcare studies I mentioned. And within these clinical trials, the technology is available and the patient get measured. But yeah, of course, I mean, whenever there is an indication, there is the opportunity to get such devices.
Yeah, thank you so much. I would like to address one more question. They keep moving because people upvote them, so I keep losing track sometimes, but I'm just going to pick one here. Okay, I'm just going to read it out. Could we say that everybody produced some kind of microclots and that the body tidies this up in normal circumstances? But when a person with long COVID overexerts, the clots are being formed and the body cannot really clear them? Is it?
Yes. So I think, the major task of blood is, besides transport of nutrition and oxygen, obviously, is clotting to prevent bleeding, right? So that is what the blood always wants to do. And also shear forces are known to induce this clotting. The body prevents, has a very, very tight regulated system to regulate that. And that seemed to be destroyed in at least a subset of long COVID people, yeah.
Do you think this could explain post-exertional malaise? The last question.
Yes, of course. I mean, I think as more clots you have, and as I showed you, right, so we have 10 microliters of blood, which we need for our measurement, but we do not measure 10 microliters. What we measure is 0.5 microliters. So basically a very, very tiny amount in total. And even there, we count up to 100 clots in some of the patients. And if you imagine that these clots are really big, right? So I have shown you in our channels, they are 20 micron high. So they can be super big, much bigger than the capillaries. And that will basically stiffen the blood and make the blood more viscous and therefore less fluid. And you can clearly see a reduction in flow. And that can be, of course, a causal link to some of the symptoms, such as PEM.
I think that was a very interesting closing statement and remark. Unfortunately, this session has come to an end. Thank you so much, Dr. Martin Queter, for your time and your sharing your insights and data. I think some people were wondering, the unpublished data you have been sharing, are they allowed to share screenshots? Can you quickly elaborate to that?
Can I prevent you from not doing it now, actually?
Okay. Okay, thank you so much. Have a wonderful day.
Yeah, thank you for the invitation again.
Thank you.